Hi, Dr. Wang here. Today I'm doing a tutorial for the practice exam for the midterm bioinfo portion. The first question is a gene bank question. So I'm copying the accession ID, including the version number .1. And here, searching for gene bank to go to NCBI gene bank. Then I'm going to paste the accession number and then search. So if this take, oh, okay, it's pulling up. So the question asks for which organism is the gene isolated from. So for this, you can search for source and somewhere down here under the features, you can see the organism right here. So this is the organism for this gene. So back to question number 2. This gene bank record happens to be identical for question 1. This may not be the case for the actual exam, so please treat every question during midterm as a fresh question, starting by pulling the gene bank record. So here I'm going to take a shortcut because I'm still looking at the same record. The question asks for how many exons. Exon is the region of the gene that ends up in the mature messenger RNA. Also, under features, you're going to see hyperlinks. That's exon. So if you double click on exon, it's going to open up a fresh tab at the bottom of your screen. So here I'm going to double click. So as you can see here, it opened up a fresh tab at the bottom. I'm going to bring this up a little bit. So here, a region of the gene is highlighted. This is the first exon. And then I can scroll through all the exons, right? So here, this is the total number of exons, right? So the second number down here, this is the total number. Coming back to question number 3. So which chromosome? So for this, we are going to come back to under feature source. So here, this is the number of chromosomes where this gene is located. Question number 4. So how many base pairs? How long is this gene? Is this sequence? So here it's right also under the source. So you can see that there are lots of information under this source tab, right? So organism, the length of the DNA sequence, and then chromosome location and so on and so forth. So this is how to solve the first 4 questions. Now let's look at question number 5. So here we have a sequence, DNA sequence, and it asks for which organism is this gene isolated from. So for this, we're going to do BLAST. So here I'm going to search for BLAST. So over here, you can either do nucleotide blast or protein blast. So our sequence is GATC, so this is a DNA sequence. So I'm going to copy the entire sequence. I'm scrolling left to right to make sure that I have copied the entire sequence. And then nucleotide blast, pasting the sequence into the search box, and then come down here, click blast. So this is going to pull out the BLAST result, searching for existing gene bank record that share sequence similarity to your input sequence. So I'm going to click on the first hit. So over here, you can see here the sequence is identical to the input sequence, right? So here you can also click on this gene bank record, and then the organism is again under the source, right? So if you go all the way down to feature source, here is the organism. All right, so coming back to question number 6. So here we have a sequence. The question asks for the longest open reading frame. So again, I'm going to copy the entire sequence, make sure I copy from the beginning to the end. And then over here, I have my APE open. So I'm going to paste the sequence in here. I'm going to make this window a little longer. All right, so to search for open reading frame, it's really important to put your cursor at the very beginning and then go to ORF at the top, click Find Next. This is going to find the next open reading frame after my cursor. So uh, not the entire gene is highlighted. So there is a possibility that there's potentially another ORF that's longer than the current one. So I'm going to hit Find Next a couple of times, right? So basically going through all the open reading frame that exists in this stretch of sequence and then look for the longest. So now I'm coming back to the first one. So this is clearly the longest open reading frame. So here with this highlighted, I'm going to go back to ORF and then translate. Over here, choose the one-letter amino acid code and then click OK. So here, this is the protein translation. So the number of amino acid is right here, right? So the number of amino acid in this are coded by this longest open reading frame. So question number 7, here we have a sequence. So here I'm again going to copy paste this sequence into my APE. So I'm going to delete the previous sequence, paste in the new one. Let's look at the question. So if you use two primers, you can amplify a product of how many base pair? To do this, we're going to first locate the two primers. So I'm going to do primer 1 first. So copy the sequence and then over here search for it. You should be able to go to under edit, find, paste the sequence here, and then find next. So here we're going to compare this two sequence. So you can see the input sequence, primer 1, is identical to the top strand. Therefore, this is a forward primer. So I'm going to make this a feature. So with primer sequence highlighted, go to feature, new feature, and then this is primer 1. So I'm just going to call this 1. And this is the forward primer, so I'm going to leave this box unchecked. All right, so you can see it added a feature that is going from 5 to 3, similar to the top strand. So this is the forward primer. Now I'm going to do this for the reverse primer. Right, so here, search again. So this is the primer 2. So here, if you compare the top sequence, top strand sequence with the input sequence, they're not identical, which means the sequence is located at the bottom strand. So you can actually see our CAG, right? So the bottom strand here is CAG. So this is a reverse primer. So here, I'm also going to add this as a feature. And then this is primer 2. And over here, I'm going to click the reverse complementary checkbox so that this is added as a reverse primer. So now to figure out how long is the PCR product, I'm going to highlight a region starting from the beginning of the forward primer and ending with the end of the reverse primer. So this is what the PCR product looks like. So I can copy this stretch of sequence and then paste this in the new window. So here, if we look at this new sequence, we can see the PCR product start with the forward primer and with the revert primer. And then over here, this is the length of the entire PCR product. Oops. All right. So now coming back to question number 8. So if you digest the sequence above with restriction enzyme, you will recover how many fragments. So over here, I'm also going to copy the sequence. By the way, during the exam, please treat each question as a fresh question. So this is why I'm copying the sequence to my APE every time. So clear the old sequence and then paste the new sequence in here. So to do digestion, simulated digestion, we're going to go to enzyme, then click enzyme selector. So the enzyme we're looking for is NLA3. So NLA, so let me search for that. NLA followed by Roman numeral 3. So I'm clicking on this to highlight the enzyme. There is a parenthesis 4, meaning this enzyme cuts the DNA 4 times. So for a linear molecule, by the way, if we don't tell you the DNA is circular, the default is linear. So most of the DNA that we gave you in the exam are linear. So this is a linear sequence. If you cut it 4 times, you're going to get 5 fragments. So you can also do a simulated digest by clicking the digest button and see those 5 fragments on the simulated gel. So this is the first fragment or the largest. Here, the next one, right? So all the way to the smallest fragment. So the size of the fragments are also summarized in the table over here under the size column. So here are how many base pairs are those 5 fragments. So the next question actually asks for the longest fragment after digestion is how many base pairs. So this is the largest number out of the 5. So in this practice exam, the sequence happens to be the same. So I'm not redoing the digestion. But during the actual exam, please redo the digestion with a fresh sequence because they might be different. All right, so now coming to question 10. So here we have 2 sequences. Those are not ATGCs, so we're looking at protein sequences. So the question asks us to identify the mutation. Mutation is defined there are 3 components to how usually we annotate mutation. The first letter is the amino acid in the wild type. The last letter is the amino acid in the mutant sequence. And the number in the middle is the location of this amino acid. So to do this, we're going to open up the alignment function. So I'm going to close a few older apps. Right, so here I'm searching for Cluster Omega. So this is an online sequence alignment tool. So over here, the default is protein, which is what we need here. So I'm going to come over here, copy the 2 sequences in. So please include the gene name, right? So greater than sign followed by the gene name. This is the FASTA format. So I'm copy both sequences into the box. Let's double check. Yeah, I have the wild type and mutant sequence pasted in. So now please use cluster with character counts because we need to know the position. So then submit. Okay, so by the way, during the exam, if this is taking too long, so just let the program run and then you can also always go to the next question. Okay, so it looks like I'm experiencing some error message. All right, so let me pause the recording here. All right, so not sure what happened. I rerun the job and they went through. All right, so if any unusual output, you can always close the tab, reopen the tab, and resubmit your sequence alignment request. So over here are 2 protein sequences aligned. The first line or the top line is from the wild type, and then the second line is from the mutant, right? So this is the beginning of the protein sequence, this is the end. So when amino acids are identical, you're going to see an asterisk underneath. So we're scanning the sequence, looking for a character that is not asterisk, which is right here. So here we have the letter amino acid in the wild type. At position, this happens to be 60, right? So here is the amino acid count. So if it's not the end of the sequence, you probably need to count a few amino acid, right? So for example, this E over here would be 61, 62, 63. So here, the amino acid and the mutant is right here. So you can see that here, this is one amino acid mutated to another amino acid at position 60. So this is the answer for question number 10. So question number 11, we have two sequences, wild type, mutant. So here ATGC, so we have DNA sequences. So here, align the two sequences above and then to distinguish between these two sequences, which of the following enzyme will cut them differently? So for this, we need to align the two sequences to find out where the mutation is. So here, I'm going to clear my old sequence, right? So this is my, okay, so since I have two sequences, I'm going to open two sequence window. So for the first sequence, I'm going to copy paste the wild type sequence. Okay, so here's the wild type sequence. I'm going to copy paste that into my new DNA window, and then coming back to copy the mutant sequence, I'm pasting that into new DNA 1 window. With the two sequences open, I'm going to go under Tools and Align Sequences. So currently, the reference sequence is new DNA. So I'm going to click new DNA 1 so that I'm comparing between new DNA and new DNA 1. So here, okay. So this opens up the sequence alignment, and we can see that the mutation is right here, right? So this is G mutated. I can't see that very clearly. It looks like a A to me. All right. So to look for restriction enzyme that cuts the sequence differently, I'm going to open another app called NEB Cutter. All right, so because I need to do this for both mutant and wild type, I'm going to open up two NEB cutter window. So in the first window, I'm coming back to my sequence alignment. All right, so over here, we also have the, let me look at this, new DNA. Oh, okay, so new DNA is on top, right? So this is new DNA sequence. So the first strand comes from the new DNA. And then the strand at the bottom here, this is the new DNA 1. So mutation is right here. So what I'm going to do is to copy probably around 10, 10 nucleotides, 10 to 20 nucleotides, right, with the mutation right in the middle. So I'm first copying this from new DNA, which is the wild type, and then pasting that into NEB cutter. So I copied all the way from CAA to GAA. Coming back to the alignment, I'm going to copy exactly the same sequence, also from CAA all the way to GAA. Right, so, and then pasting that into my second NEB cutter window. So just confirm that I'm copying the same range. Yeah, that looks about right. So here I'm going to submit both jobs. All right, so now I'm going to put this two side by side. So here you can look at both window. So now you can clearly see, right? So even the two sequences are pretty similar to each other, except right in the middle, we have a G over here, and then we have a C over here. So this one change in nucleotide actually resulted in different patterns for restriction enzymes. So over here, you can see a lot more enzymes that are cutting this DNA, but not cutting this DNA. So among all the unique ones, right, that are present in this window, one of them is a choice here, right? So here are 5 choices. So one of them is an enzyme that only cuts this molecule, but doesn't cut this molecule. So this is the answer for question 11. So now let's come to question number 12. So over here, this also requires APE, so I'm going to clean up over here a little bit. So here, this is my DNA window. As usual, I'm going to clear that so that I can put my new sequence in here. Okay, so here the question says there is a mutation, right? So highlighted in red, and it's at position 54. So in this case, because it's given the location, I can actually just go to 54 directly. All right, so 54 is about here. And another way to find this particular position is to do a search find. Right, so if I look for it, you're going to see that it highlighted basically this A over here. So I'm going to go to feature with this mutation highlighted, call this mutation, so that I'm marking this on my strand. So down here are 4 primer sequences. So I'm going to look for them in my sequence. So this is primer A. So let's compare. Primer A is not identical to the top sequence, right? Top sequence is CGG. Here it's TCA. So this means primer A is a reverse primer. So I'm going to feature, new feature, and then this is primer A, and clicking the reverse complementary checkbox so that I can add primer A as a reverse primer. So now I'm going to do the same for primer B. Copy the sequence, search for it, and then decide whether this is, determine whether this is on the top strand or bottom strand. All right, so GCTA, identical to the top strand. So this is a forward primer. So I'm going to go to features, new feature, and then call this a forward primer. Now let's do the same process for primer C. So primer C, TTCTT, all right, so not identical, right? So which means this is a reverse primer. So here I'm going to go to feature, add this as primer C and as reverse primer. So here C is added. Now let's do the last one, primer D. Okay, so primer D is identical to the top strand sequence. So this is a forward primer. So here, adding this as primer D, leave this unchecked so that it's displayed as a forward primer. So now here are everything added. So I'm going to open up all the sequences. You can see everything. Mutation is right here. So we're looking for a pair of primer that that with the mutation in the middle. Right, so this is how PCR works. If we amplify a PCR product, the mutation has to be in the middle of this PCR product. And we're also looking for a pair of primer that are running towards each other. So the sort of the sequence we're looking for is forward primer first, followed by the mutation, and then followed by the reverse primer. And then that is the answer for question number 12.